This is Group 2 with Jarek, Robert, Stephen, and Daniel. We'll be presenting on ADH and alcohol tolerance.
In this experiment, we will be studying the effects of a mutation in a single gene, the gene that codes for the enzyme alcohol dehydrogenase, or ADH. ADH catalyzes the following enzymatic reaction that converts alcohol into acetaldehyde. Right. This is the reaction in which alcohol is catalyzed. You can see this is ADH, which is the enzyme. Ethanol is right here. It is the substrate. And NAD acts as the electron carrier. So what happens is ADH produces alcohol, which turns it into acetaldehyde. And the reduced electron is transferred to the electron carrier, electron acceptor called NAD, which turns it into NADH. So that is the reaction. And for this experiment, we chose to study the ethanol sensitivity of two different strains, a range of fruit fly called Drosophila melanogaster. We chose the fruit fly, these Drosophila melanogaster, because these fruit flies have been used as model organism in genetics for many years now. They have short life cycles, which it's quite easy to maintain. And their genetics is very, very well understood. We picked fruit flies that carry two different strains of ADH, as I mentioned before. One is a wild type, which has the gene that catalyzes for the production of the enzyme ADH, and one gene catalyzes does not have the ability to produce that enzyme. We call the gene that has the ability to produce that enzyme ADH plus, and the one that doesn't have it as ADH negative.
For our equipment and materials, we used 12 fruit flies, 4 containers labeled as ADH positive and ADH negative for alcohol and H2O, 4 cotton balls to soak in the alcohol and water, as well as 4 stoppers, and our ethanol, which is this fine wine over here.
The ADH negative group of fruit flies lacks the ADH gene that codes for the breakdown of alcohol. We hypothesized that this sample group will have a low survival rate.
Here's a table representing the class data at our starting time and after 24 hours. At the starting time, we started with 31 and 28 ADH plus and negative flies in alcohol, and in water we had 21 ADH plus and 31 ADH negative flies. After 24 hours, there were 26 ADH plus flies and 0 ADH negative flies in alcohol. In water, there was 19 ADH plus flies and 27 ADH negative flies in water. From the table, we can see that the flies that did not have the protein that synthesized ethanol died, and the ones that did survived after the 24-hour period. This graph represents the percent of flies that survived after a 24-hour period. 90% of flies in ADH positive survived in water, and 87 flies in ADH negative survived in water. In alcohol or ADH positive flies, only 84% survived. Our ADH negative flies, 0% survived. Again, concluding that the flies mutated for ADH were not able to synthesize alcohol, causing them death. During time intervals at 15 minutes, 30 minutes, and 1 hour, we did not see any deaths or any changes in fly survival rate. But at 2 hours, in ADH negative fly in alcohol, we did see 1 death. In the flies that were in alcohol, whether ADH positive or ADH negative, both of their behaviors varied compared to the flies in water. They moved around more, and they were more clumsy. We noticed that the flies in alcohol tended to cluster into groups compared to the flies in water, which were more spread out. You can see from the data that the flies that were defective for the ADH gene in alcohol did not survive, supporting our hypothesis with the same conclusion. For question 1, a ADH gene that is defective cannot produce the enzyme that synthesizes ethanol, meaning that this person may be more prone to alcoholism because the alcohol stays in their system for a longer amount of time, making their body depend on this drug. This is based on science, but it's not sound science because we do not know if it's just the ADH gene that plays a part in alcoholism or if it's many other genes as well. At this point in history, this law is not constitutional. We cannot force someone to make changes to their body. This law does not solve all the problems for drunk driving, for there may be people as well who do have the ADH gene that's not defective that still drink and drive. The only way this law would be enforced if we were to force it on newborn children. It would be the same of choosing whether to circumcise or not circumcise. Instead, it would be to scan the genome of a specific person to see if they have a defective ADH gene or not. I would not be in favor for such a law because we do not know how the genes affect an individual. For question 6, I would not be in favor of a similar law for predisposed pedophilia or violent behavior because we do not know if the gene will ever be expressed and whether an individual will actually act. For question 7, I personally would not care if someone was genetically predisposed to infidelity because again, I do not know whether their mind is stronger than the gene. My fellow lab partners may disagree with that.